Hi students, I'm Geeta, Botany faculty. Today I'm going to teach you the topic of cell cycle, mitosis. Let us see what is cell cycle. When a cell is dividing into two daughter cells or maybe four daughter cells, the type of division which is taking place in the cell cycle that you are going to learn today. So let us see the first one, the cell division. You know, cell cycle is divided into interphase and M phase. You are going to learn today the M phase, that is divisional phase, and particularly in that mitosis. Let us see that mitosis, what is going to happen in the mitosis or M phase. The first one, mitosis is nothing but when a parent cell is dividing into two daughter cells. That is going to happen in the mitosis. So, the mitosis or M phase is the most dramatic period of the cell cycle. So much drama will be happening in the cell when a cell is dividing. So M phase or mitotic phase is considered as most dramatic period of the total cell cycle. So in interphase, less drama is going to happen, and in M phase, so much drama in terms of the cell chromosomes are moving towards the ends and the daughter nuclei formation, the chromosome splitting, and then finally the cell will be dividing in terms of cytoplasm, in terms of nucleus, like that. So much drama is happening in this cell cycle, particularly in M phase. So it is called most dramatic period of the cell cycle, where all the components of the cell are actually reorganized. All the components will be reorganized. That is why it is called as most dramatic period of the cell cycle, involving major reorganization of virtually all components of the cell. And in this M phase is also called as equational division, because whatever the parent cell contents are there, like chromosomes, they will be equally distributed to both the daughter cells. And whatever the cell organelles are there in the parent cell, they are also equally distributed to the daughter cells. As everything is distributed equally to both the daughter cells, this is called as equational division. So mitosis, another name is equational division, because everything in the cell will be equally distributed to both the daughter cells. Since the number of chromosomes in the parent and the progeny cells is the same. See, for example, if you take the human cell, our cells, we are having diploid number 46 chromosomes. So if that cell is undergoing the mitotic division, the formed daughter cells will be having the same 46 chromosomes only will be there in the daughter cells. So when the chromosome numbers are same between the parent and progeny cells, it is called as equational division. So it got the name equational division because of the chromosome numbers are also equal between the parent and the daughter cells. The M phase starts with the nuclear division. In M phase, we have two steps are there. First step is called karyokinesis, which includes nuclear division. And second step is called cytokinesis, which includes the cytoplasmic division. So first one is the nuclear division, corresponding to the separation of daughter chromosomes, where the daughter chromosomes are formed and the daughter chromosomes will get separated and two daughter nuclei are formed in the karyokinesis end. And then it usually ends with the division of the cytoplasm, which is called as cytokinesis. The division of cytoplasm is called as cytokinesis. First M phase will be having the first step called karyokinesis, which is nothing but nuclear division. One parent nucleus divides into two daughter nuclei, and in that the chromosomes also are getting separated. The second step is called cytokinesis, which is nothing but division of cytoplasm, where one parent cell is dividing into two daughter cells. That is going to happen here. Let us first begin with the karyokinesis. Karyokinesis is nothing but nuclear division, and this karyokinesis involves four stages. The first stage is called prophase, second stage is called metaphase, and the third stage is called anaphase, and the fourth one is called as telophase. So in karyokinesis, we have the four phases, four stages or four subphases are there. They are called as prophase, metaphase, anaphase and telophase. Then this is the prophase. When a cell is entering into the prophase, okay, this is the early prophase. You have got that NCERT pictures like this only. They might give you these pictures and they'll ask you which phase is this. When you see this blue color chromatin and red color chromatin, inside the nucleus, then it is the early prophase. Okay, you cannot see this clearly. The chromosome is having actually two chromatids. The blue one is actually having two chromatids, like they are all intercoiled or intertwined. They are intertwined and you can't see the chromatids separately in this stage. So when you can see the chromatin material, okay, which are intertwined, that is nothing but early prophase. You can see here, this is the late prophase where this type of chromatin, it has organized, it has become a compact structures. They are called as chromosomes. These are now called as chromosomes. This is called chromatin or chromosomal material. So in the early prophase, the chromatin material will be there, like elongated threads and intertwined threads will be there. And in the late prophase, this type of elongated chromatin, it would have become compact, short, rod-shaped structures called chromosomes. So these are the changes. First, they will happen in the prophase. By the picture, you can identify that. And in the same picture, you can see this is the nuclear envelope. Nuclear envelope is there. This nuclear envelope, it is complete here, but when you see this late prophase, this nuclear envelope is ruptured or it is broken down. The nuclear envelope is broken down and it is not that clear. That means in the late prophase, nuclear envelope disappears. It will be disappearing. Okay. So here you can see clearly that nuclear envelope is getting broken down and it will disappear in the prophase. Let us see the main events of the prophase. In the prophase, first chromosomal material condenses to form the compact mitotic chromosomes. That chromatin, this elongated chromatin will be condensing together to form the short and compact chromosomes. But here you should remember that all the chromosomes will be having two sister chromatids which are actually formed in the S phase, synthetic phase of the interphase. And in S and G2 phases, already two chromatids are there for each chromosome. Two chromatids per one chromosome. And they are actually connected at the centromere. But we cannot see that clearly because they are all intertwined here and we can't see them clearly. Chromosomes are seen to be composed of two chromatids attached together at the centromere. So when you look at the structure, they will be attached at the centromere like this. These two are called as sister chromatids. And this structure is called as centromere or primary constriction. So the two chromatids are actually formed in the S phase and in G2 phase it will be continued like that. But as they are not condensed, we can't see them clearly and both of them are, that sister chromatids are intertwined. So we can't see them clearly in the early prophase. And in the late prophase, they will become compact structures and there you can see little bit clarity with two sister chromatids joined at the center with the help of centromere. That thing is going to happen in the prophase. Next one, after that, the centrosome. You know that centrosome is a particular cell organelle which is present in the animal cells. That centrosome also got duplicated in the S phase only. So instead of one centrosome per animal cell, we will have two centrosomes per animal cells after the S phase. So that centrosome which had undergone the duplication during the interphase, particularly in the S phase, begins to move towards the opposite poles of this cell. That centrosome will be moving, two centrosomes will be there, one centrosome at the one pole and another centrosome will start migrating towards the opposite pole of this cell. So that event is the second event. First event, condensation of chromatin into chromosomes. Second event is centrosome will be moving towards the opposite pole. The third event, each centrosome radiates out microtubules called asters. Each centrosome will be having this type of structure with two centrioles and they start radiating out, giving out some kind of microtubules and it looks like star-shaped structures. So these are called asters. And this type of centrosomes, two will be there in the cell, and the second centrosome will be starting moving towards the opposite pole. So it will start giving rise to the microtubules. So two centrioles with their spindle fibers will form the asters. These asters are seen only in animal cells. Only in animal cells. In plant cells, these are absent. And that asters are formed in the animal cells, and the two asters together with spindle fibers form the mitotic apparatus. So in the cell, what is going to happen? Two asters will be there, one centrosome and second centrosome, and then microtubules will be there. So two asters per animal cell will be there, and the two asters along with the middle spindle fibers if the spindle fibers are there like this, okay, these things together called as mitotic apparatus. So two asters and the spindle fibers in between them will be together forming the mitotic apparatus. These are the major events which are going to happen in the prophase. Then the fourth one, cells by the end of the prophase. Okay, by the end of the prophase, when viewed under the microscope, they do not show Golgi complex, then endoplasmic reticulum, and then nucleolus and nuclear envelope. When you look at the cell in the previous diagram, you have seen that nuclear envelope is broken down. Not only nuclear envelope, okay, nucleolus, a rounded body which is present without any membrane, that is nucleolus. It is present inside the nucleus. That nucleolus, then Golgi complex, then endoplasmic reticulum. These four will start disappearing in the cell. So the first thing in the prophase is condensation of chromatin material into compact chromosomes, where each chromosome will be having two sister chromatids that are joined in the center at the centromere, by the centromere. Then centrosome present in the animal cells, it will start migrating towards the opposite poles. Then each centrosome will be giving out, radiating out microtubules to form the asters. Two asters plus spindle fibers in between them will form the mitotic apparatus. By the time mitotic apparatus is formed, then in the cell, nuclear envelope, nucleolus, Golgi and ER disappears. So all the chromosomes will be released into the cytoplasm. All the chromosomes are now released into the cytoplasm. This will be the end of the prophase. Okay, but there is no clear-cut difference between the prophase and metaphase transition because it is a continuous process. It will be happening continuously. For our convenience, we are dividing them into the perfect stages like prophase, metaphase. This is the beginning of the prophase. This is the end of the prophase, like that. But you have to remember one thing, that it is a continuous process. Okay. So by the end of the prophase, the chromosomes got released or scattered into the cytoplasm as nuclear envelope disappears. That is the ending of the prophase. Then the next phase. So you can see here the two asters are there. This is one aster. This is one more aster. Okay, so two asters are there, and these are spindle fibers. So now together it is called as mitotic apparatus. This is the transition stage to mitosis. You can see that transition to metaphase. Okay. Then this metaphase characters will be there. Now the metaphase will be continued after the prophase, the continuity. So in the transition, you can see that chromosomes are released into the cytoplasm. Asters, second aster already it got moved towards the opposite pole. Spindle fibers are coming. Okay. And mitotic apparatus is formed, nuclear envelope disappears, and all the chromosomes are released. That is transition to metaphase. They are moving. You can see that chromosomes are scattered, spindle fibers are getting formed. In the metaphase, what happened? Asters would have reached already the opposite poles. And now the spindle fibers coming from each pole, they will be connected to the centromeres. Each chromosome, it will be having in the center centromere, because two sister chromatids are connected. Already I told you two sister chromatids are there. They are connected in the center by the centromere. But on both the sides of the centromere, we have two disc-shaped structures will be present on the surface of the centromere. These are called kinetocores. Kinetocores or chores. So, two will be there, disc-shaped structures on the surface of the centromere. Now, from here, the spindle fibers will be coming out and they will be connected to the kinetocores from both the poles. From one aster, from this side one aster, they will be coming and they will be connected to the kinetocore, and from the another aster, one more spindle fiber will come and it will be connected to this spindle fibers. So now you know that each chromosome will be having two sister chromatids and they are connected at the center by the centromere, and on either side of the centromere we have two kinetocores. They serve as attachment point for the spindle fibers. So from both the poles, okay, each chromosome is connected from both the poles by two spindle fibers. From this pole, one spindle fiber will be coming from this aster, and from this aster also one spindle fiber will be coming, and by two spindle fibers each chromosome is connected from both the poles. This is the important thing here. So from one aster, one spindle fiber will come and to that side kinetocore it will be attached. From another side, one more spindle fiber will come and to the same chromosome on the opposite side to the other sister chromatid kinetocore, this will be attached. So the attachment of kinetocores, okay, to the spindle fibers or to the spindle fibers, that will happen here. So spindle fibers will be coming from both the poles and they will be connected to the kinetocores. After this attachment, okay, these spindle fibers will undergo contraction and relaxation and they will bring the chromosomes, all the chromosomes of that cell to the center of this cell. This is called equator of this cell, equator. So because of the contraction and relaxation of the spindle fibers from both the ends, this side and this side, all the chromosomes will be aligned in the center or equator of this cell. This plane of alignment of chromosomes in the equator or in the center of the cell is called as metaphase plate. So now you can see this is the metaphase plate. One metaphase plate is formed here. And in this metaphase, what you have to remember, first event what is going to happen is the spindle fibers from both the poles will come and they will be connected to the kinetocores of one chromosome. So two spindle fibers will come and connect to the two kinetocores of one chromosome. First event. Second event, by the contraction and relaxation of all these spindle fibers in the cell, all the chromosomes, how many chromosomes are there in that cell, all of them will be coming and aligned in the center or equator of this cell. This plane of alignment of all the chromosomes in the center or equator of the cell is called metaphase plate. Because this plate is formed in the metaphase, it is named as metaphase plate. Okay. Then let us see the events what is going to happen in the matter. The complete disintegration of the nuclear envelope marks the start of the second phase of the mitosis. The beginning of the metaphase means it is indicated by the complete disintegration of the nuclear envelope. Nuclear envelope is completely disintegrated. That is the beginning of the metaphase. Condensation of chromosomes is also completed and they can be observed clearly under the microscope. Condensation started in the prophase, now it is also completed. Now we can see clearly under the microscope the chromosomes. Then this is the stage at which the morphology of the chromosomes is most easily studied. This is the best stage to study the morphology, structure of the chromosome, because we can see very clearly centromere, two sister chromatids, every part of the chromosome we can see. So the structure of the chromosome or morphology of the chromosome can be studied in metaphase. Okay. Then metaphase chromosome is made up of two sister chromatids and they are held together by a centromere. Already you know that two sister chromatids are there. They are actually formed in the S phase and G2, and now those two sister chromatids are joined at the center by the centromere. Then small disc-shaped structures at the surface of the centromeres are called kinetochores. Kinetochores are present on the surface of the centromere on both the sides. So one centromere will have two kinetochores. And these structures serve as the sites of attachment of the spindle fibers. So they are the points of attachment of spindle fibers. And that kinetochores are also formed by the spindle fibers only. And they will connect the chromosomes, okay, that are moved into the position in the center of the cell. So spindle fibers will come and they attach to the kinetochores and they will be moved to the center of the cell. Then the spindle fibers attach to kinetochores of the chromosomes and they will move the chromosomes to the center. Chromosomes are moved to the spindle equator and get aligned along the metaphase plate through the spindle fibers to both the poles. So each chromosome is connected to both the poles by two spindle fibers. Because we have two kinetochores, so two sides it will be connected. The plane of alignment of the chromosomes at metaphase is referred to as metaphase plate. One plate is formed because of the alignment of all the chromosomes in the center or equator of the cell, because the spindle fibers are connected. So this is the end of the metaphase. And the cell at the end of the metaphase, it will be looking exactly like this. So from this pole, one spindle fiber will come and attach to the kinetochore of this chromosome, and from this pole, one more spindle fiber will come and attach to this kinetochore. So two sister chromatids are joined to the two spindle fibers from both the poles. Then the third phase is the anaphase. In this, what happens is, the spindle fibers which are connected to the kinetochores, they will undergo contraction and they will be pulling the chromosome from both the sides, because one chromosome is attached to two spindle fibers. So these spindle fibers will contract and they will be pulling the chromosome completely, half in one direction and half in another direction. Because of that increased pressure, the centromere splits. The centromere splits and two sister chromatids will get separated. Now the sister chromatids, each sister chromatid will be having this type of structure. Now this type of sister chromatids are getting separated because of the splitting of centromere. Now these structures are called as daughter chromosomes. Daughter chromosomes. They are formed in the anaphase. Because of the increased pressure on the centromere from both the sides, the centromere splits and it results in the formation of sister chromatid separation, and it forms the two daughter chromosomes. One chromosome will be forming the two daughter chromosomes. Then the spindle fibers will start undergoing the contraction, pulling nature. So when they are moving, first the centromere will be moving towards the pole first, latter followed by arms behind. When they are moving, they will be moving. First centromere will be going first and then arms are trailing behind. Arms will be going behind. Always first centromere will be moving, leading edge, and then behind arms will be going. That is called trailing end, trailing edge. So when they are moving in the opposite direction, they will be taking the shapes of V, L, J and I shapes. So anaphase is the best phase to study the shapes of the chromosomes. So metaphase is the phase where we can study the morphology of the chromosome or structure of the chromosome. Anaphase is the phase where we can see the shapes of the chromosomes like V, L, J, I. Okay, when they are moving towards the opposite poles, okay, the daughter chromosomes, we can see the shapes of the chromosomes. Now let us see the events of the anaphase. The matter, centromere split. This is the first event. First centromere split and chromatids get separate. The daughter chromosomes of the future daughter nuclei begin their migration towards the two opposite poles. These are the daughter chromosomes and this started moving towards the opposite pole, this started moving towards the opposite pole. When they are moving, you can see that centromere is going first, followed by arms. Always centromere will be going first, followed by the arms behind. So leading edge is always centromere and trailing end are always arms. So in this way, the daughter chromosomes will migrate towards the respective opposite poles. As each chromosome moves away from the equatorial plate, the centromere of each chromosome remains directed towards the pole and hence at the leading edge, and with the arms of the chromosome trailing behind. As I said, centromere will be going first and followed by the arms behind. So these events will happen: centromere splitting, sister chromatids separation, daughter chromosomes formation, and daughter chromosomes will migrate towards opposite poles. When they are moving, centromere will go first and then arms will be following that. Leading edge will be centromere, trailing edge will be the arms. When they are moving, we can see the shapes of the chromosomes like V, L, J, I shapes. The next one, telophase. This is the last stage of the karyokinesis where the events of prophase will be exactly opposite in the telophase. You know, in the prophase you have learnt that condensation of chromatin material occurs, chromosomes are formed. Here what happens? This type of chromosomes which are formed, they will undergo decondensation. They will become the chromatin. Chromosomes will become the chromatin. It is called as decondensation. Decondensation. So decondensation of chromosomes into chromatin occurs in the telophase, which is exactly opposite to the prophase. Then here the prophase events are like nuclear envelope, nucleolus, Golgi complex and ER. They disappear. Here nuclear envelope, this is the nuclear envelope that is getting reformed. This nuclear envelope is reformed because already chromatin material moved towards the opposite poles. They migrated already by the end of the anaphase only. So now at the poles, chromatin material is formed. After that, the nuclear envelope reforms. And then in the cell cytoplasm, Golgi complex, Golgi, ER, they are reformed again. So these events are exactly opposite of prophase. So we'll say that prophase is exactly opposite of telophase. Let us see the matter. Chromosomes cluster at the opposite spindle poles and their identity is lost as discrete elements. Can you see in this diagram any chromosome identity? Can you tell me? We can't. So they are losing their individuality, they are losing their identity as separate elements like this. Okay. Then the nuclear envelope develops around the chromosome clusters at each pole, forming two daughter nuclei. So once the nuclear envelope is formed, this total structure is now called as daughter nucleus. How many daughter nuclei are formed here? Two daughter nuclei are formed in the telophase. Then the nucleolus, Golgi complex and ER are reformed or reappearing. They are reformed here. So all these three events are exactly opposite to that of the prophase. So it is exactly opposite. Okay. This is the end of the karyokinesis. The next stage is the cytokinesis, where a cell will be dividing its cytoplasmic contents. Nuclear division happened. Already we got two daughter nuclei by the end of the telophase. Now cytokinesis will divide one parent cell cytoplasm into two cells, two daughter cells. Cytokinesis is nothing but division of cytoplasm. In animal cell, it happens in a different method. In plant cells, it occurs in a different method. In animal cell, cytokinesis will happen by the end of the telophase only. It will start. And this structure is called as cleavage furrow. In animal cells, it occurs by the cleavage furrow method. One furrow is actually formed in the middle of the cell. And the plasma membrane will constrict in the center of the cell. The plasma membrane of the parent cell, it will be constricted exactly in the center like this. So the plasma membrane will be constricting in the center of the cell. One invagination type, it will be appearing in the center of the cell and that grows towards the center. that will be growing towards the center. So once it grows towards the center, the cleavage furrow is formed, and the cleavage furrow, okay, it is formed towards the center like this, centripetal manner, and the cytoplasm of this parent cell will be dividing into two. And you know, all the cell organelles also will be distributed equally to the daughter cells. So in animal cells, the cytokinesis is achieved by the appearance of a furrow in the plasma membrane. You know, in the animal cell, this is the plasma membrane only, not the cell wall. Cell wall is present in plant cells, but in animal cells, plasma membrane is there. So plasma membrane will show invagination exactly in the center of the cell to form the cleavage furrow, and which grows, deepens gradually towards the center, and it divides the one parent cell into two daughter cells. The furrow gradually deepens and ultimately joins the center, dividing the cell cytoplasm into two cells. So one parent cell, it got divided into two daughter cells. What you have to remember here in the animal cytokinesis is the cleavage furrow will be growing in the centripetal manner. It grows centripetally. This is the important word here. Always furrow or cleavage furrow will be growing towards the center. So centripetal growth will be there. And then it divides the single cell into two daughter cells. This is what is going to happen in the animal cells. Next one, plant cell cytokinesis. In plant cells, it is little bit complex, the events of cytokinesis, where first the Golgi vesicles, these are called as Golgi vesicles. Vesicles will start accumulating in the center of the cell. First one vesicle will come and it will be appearing in the center. First one vesicle, then two vesicles will come and they'll exactly occupy the center of the cell. Then that vesicles will be growing towards the ends of the cell. They'll fuse and they'll be growing towards the ends of the cell. When they're going towards the ends of this cell, okay, it will be forming one barrel-shaped structure. You can see that this is looking like a barrel-shaped structure. This barrel-shaped structure is called as phragmoplast. Structure called as phragmoplast is formed. After that phragmoplast will be fused together. Now it forms the cell plate. Now it is called as cell plate. That phragmoplast, all of them will be fused and it grows from the center towards the side and it forms the cell plate. Cell plate represents the middle lamellum. It is a precursor actually for the middle lamellum of the cell wall. You know, the middle lamellum is a cementing layer of the cell wall which connects the two neighboring cells together. So that cell plate will be formed after the phragmoplast. And cell plate is a precursor of the middle lamellum of the cell wall. And this cell plate, how it is growing? Growing means it is growing from the center towards the side. That means it grows, cell plate grows centrifugally. It grows centrifugally. From the center towards the sides, it will be growing. So in plant cells, the cytokinesis occurs by cell plate method, which grows centrifugally. In plant cells, wall formation starts in the center of the cell and grows outward to meet the existing lateral cells. It starts in the center and it will be growing towards the existing lateral walls. The formation of the new cell wall begins with the formation of a simple precursor. One simple precursor is there, that is called cell plate. What is the precursor of the new cell wall? Cell plate. First it will start with the formation of the cell plate. That represents the middle lamellum between the walls of the adjacent cells. So that will be forming, okay, middle lamellum, and that represents the cell wall part. In some organisms, karyokinesis may not be continued by cytokinesis. So if that happens, imagine two daughter nuclei are formed, but cytoplasm is not dividing. Then two daughter nuclei are present in one cell only. In that cases, what happens? The chromosome number got doubled. So it results in a multinucleate condition, actually binucleate condition, but if it continues further, it results in the multinucleate condition. So in some organisms, karyokinesis is not followed by cytokinesis, as a result of which the multinucleate condition arises, leading to the formation of syncytium. Syncytium is nothing but multinucleate condition, which you can see very clearly in the liquid endosperm of coconut. You know, the coconut water is there, that liquid endosperm is having the syncytium condition because many nuclei are present in that. Karyokinesis happened, but cytokinesis did not happen, so it is a free nuclear endosperm. That results when karyokinesis is not followed immediately by the cytokinesis. That's all. So by the end of the cytokinesis, one parent cell will divide into two daughter cells. Then the significance of mitosis, the last importance of mitosis. In multicellular organisms, body growth occurs by the mitotic division of the cell. All the multicellular organisms like us or animals will be growing, okay, plants, they all will be growing because of the mitosis. Growth is the result of mitosis. Then replacement is also one more function of the or significance of the mitosis. Replacement of worn out tissues, okay, worn out damaged tissues or old cells or tissues, and repair out the injured tissues occur by mitosis. One is growth, another one is replacement, another one is repair. These all things are due to the mitosis. In unicellular organisms, only in unicellular organisms like euglena, amoeba, okay, mitosis is involved in the asexual reproduction. Basically, it is involved in the reproduction. So, in unicellular organisms, if you consider, this helps even in the reproduction, asexual reproduction. In plants, vegetative propagation involves only mitotic division. In the plants, like vegetative propagation is there, like stolons, suckers, okay, these are all because of mitosis. Not only this one, we have the upper layers of cells of epidermis, okay, they are increasing in their number because of mitosis. Our gut lining cells, they are also increasing in number because of mitosis. And in the plants, cambium activity is there. The cambium will be growing. Cambium is a meristematic tissue and that cambium is able to divide, okay, and it is dividing, and the division is nothing but mitosis. And you should remember that mitosis occurs always in the diploid cells only. Diploid cells only. But we have some organisms like social insects and lower plants. Like algae and fungi, it can also happen in haploid cells. Particular examples are there like drones, honeybee drones are there, haploid insects are there, okay, social insects and some lower plants like algae and fungi, mitosis will be happening in them because they are haploid cells. Generally, haploid cells will not go for that according to NCERT. So, diploid cells only mitosis will happen, and the haploid cells also it can happen, for example, social insects and lower plants. Cambial growth, differentiation, these are all the significance of mitosis. What you have to remember by the end of the mitosis means one parent cell, it results in the formation of two daughter cells exactly with the same genetic complement, same number of chromosomes and same number of cell organelles, everything will be same. That is why it is called as equational division. So, same genetic material will be there for both the daughter cells. So, the daughter cells will be qualitatively similar to the parent cell and even quantitatively they are similar to the parent cell, and they are same between them. Among them also they are same. Quantitatively and qualitatively also they are the similar cells. So, by the end of mitosis, one parent cell will divide into two daughter cells which are exactly identical, and they can further continue the division. That's all about the mitosis. Thank you.